So now we will see the characteristics or difference between cell wall of gram positive and gram negative. So first thing is the peptidoglycan layer. So peptidoglycan layer is thick. So this is a thick peptidoglycan layer, and in gram negative bacteria the peptidoglycan layer is thin. Next one is teichoic acid. Teichoic acid is present in gram positive bacteria but not in gram negative bacteria. Next thing is lipid. Lipid is, we have seen that in gram negative bacteria the outer membrane, here we can see, here we can see in the outer membrane the lipid molecules are present in the cell wall of gram negative bacteria. So the lipid molecules are present in as a lipopolysaccharide in the outer membrane of gram negative bacteria but not in gram positive bacteria. So very little in gram positive bacteria. The next one is outer membrane. So outer membrane is present in gram negative bacteria but not in gram positive bacteria. Periplasmic space is very less in gram positive bacteria because the in gram positive bacteria the peptidoglycan layer is very compact and thick, and they only have a cell membrane and the peptidoglycan layer. So the space between this peptidoglycan or cell wall and the plasma membrane is very less. This one is very less. So that's why periplasmic space is mainly absent in gram positive bacteria, but in gram negative bacteria the space between the outer membrane and cell membrane, between these two structure, the peptidoglycan layer is very less, very small. So there, in that portion the space is large or comparatively large in gram negative bacteria. So that's why periplasmic space is mainly present in gram negative bacteria. Next thing is cell shape. So as the peptidoglycan layer have a very thick peptidoglycan layer, or in gram positive bacteria peptidoglycan layer is very thick, that's why it gives a, always gives a rigid structure, but in gram negative bacteria that is rigid or flexible because their because their peptidoglycan layer is less, comparatively less in gram negative bacteria. Result of enzyme digestion. So if we digest the gram positive bacteria, then we will see peptidoglycan less gram positive bacteria, and that form will be called the protoplast. And if we treat enzyme digestion in the gram negative bacteria, then we will see the cell wall less or the peptidoglycan layer less gram negative bacteria, which is known as spheroplast. So here we will see the outer membrane and inner membrane, outer membrane and inner membrane, okay. But in protoplast we will see only the, only the inner membrane or cytoplasmic membrane, okay. So this is the main difference, that is the protoplast for gram positive bacteria, spheroplast for gram negative bacteria. Next thing is sensitivity to dyes and antibiotics. And gram positive bacteria is most sensitive because their peptidoglycan layer is high or very thick, so they are more sensitive to antibiotics which are effective to peptidoglycan layer. And the gram negative bacteria is moderately sensitive because their peptidoglycan layer is less or thin, that's why. So the main example of gram positive bacteria and gram negative bacteria is gram positive bacteria means Staphylococcus aureus, this is a one example, and gram negative bacteria Escherichia coli is an example. Okay, so move on to the next slide.